Hello, folks. Welcome to today's lecture. This channel is going to be dedicated to teaching the basic medical sciences like microbiology, pharmacology, then biochemistry, anatomy, and everything. But we are going to first start with microbiology, and therefore we'll start with general bacteriology first. And so the first topic of general bacteriology is bacterial anatomy. So without further ado, we'll just get right into it. So to understand the bacterial anatomy, we need to understand what sort of a cell bacteria is, and bacteria is a prokaryotic cell. So, what is a prokaryotic cell? We have most of us have already read this in our schools that prokaryotic is basically a primitive cell. Before all of us were born, the only thing that was on the face of this earth was just this prokaryotic cells, that is, the bacteria. Because and why it is called primitive? Because the nuclear the powerhouse, or the not the powerhouse but the center of each cell is the its DNA component or its nucleus. So the nucleus of a prokaryotic cell is very primitive and it is called nucleoid, whereas the nucleus of eukaryotic cells are better developed than this, which has like for say apart from nucleoid, it has a histone protein, then it has a nuclear envelope and a lot of other things that we'll discuss later. But this is how prokaryotic cells look like. To just review in a short bit, this is the center of the cell, or the main part of the cell, that is the nucleoid. This is the cytoplasm. It's not very much different from our cells. These are you can see these are ribosomes. You see there are two segments, like 50s and 30s. We'll discuss this in the later part of the study. These are some small granular inclusions. You can see this is a flagella. This is the cytoplasmic membrane or cell membrane. This is the cell wall, which is very important in case of bacteria. Especially, there's a component called peptidoglycan that lies within the cell wall that makes the bacteria very unique to any other cells. We'll also discuss this later in the class. And then we have a capsule. Capsule is not present in all bacteria, but it is an important structure in a lot of in some of the bacteria. We'll also discuss this later in the class. So that was a recap. So to summarize the difference between the prokaryote and eukaryote, we're not going to spend a lot of time within it. Just so you know, you see the prokaryotes are bacteria, mainly archaea or cyanobacteria, and all other bacteria that we know, all other cells that we know, like animal and plants, these are eukaryotes. The size of eukaryotes are bigger. Nucleus is primitive, whereas ours are true nucleus. We're sort of obsessive and narcissist like that. We call ourselves true, and everything else is just primitive. Nuclear membrane is present in case of eukaryotes. Chromosome, it's single, double-stranded, and circular, whereas in our case it's multiple and linear. The genome, they have single set of genome that's haploid in our case, except for the sex chromosome, it's usually diploid. And they do not have a nucleolus, whereas we have a nucleolus inside our nucleus. And the cell division is basically either we have mitotic division or meiotic division, and they basically cell divide in a binary fission. And extrachromosomal DNA and extra set of DNA, they have usually in plasmid, which is one of the components of a bacteria, which we'll also discuss later in the class. And in case of eukaryotes, we have a little bit of extrachromosomal DNA in our mitochondria. And in case of plants, it's chloroplast. Okay, to move on to the later differences, what we need to know about it is that other, the well-developed organelles in the cytoplasm, prokaryotes don't have that because it's a primitive cell, whereas like the fancy organelles like mitochondria, Golgi bodies, and endoplasmic reticulum, where we do have these fancy organelles, which the prokaryotes are lacking. In case of ribosome, it is 50 plus 30, it's 70s, and in case of ours, it's 80s. To better understand this, I'm going to show you how it basically happens. You can see that 60 plus 40 should equal to 100, right? But it is 80. And here we can see that 50 and 30 equals to 80, but here it is 70. So what actually happens? So basically, you see the ribosomes have this like two sediments. For example, you can say that it's 50, this one is 50, and this one is 30. So you can see the segment is 50 and 30, but in this middle segment, this 10 is like, you see, this segment is 50s, this segment is 30s, but they both share this 10s segment. So this is condensed, this part is condensed, and they both share this portion. So this section is mutually shared, so that's why the 10s is oblated from the total summation of 80s. So it basically, the overall, it's 70s. So that's how it gets 70s. So I hope that is clear for everyone. So the cytoplasmic reticulum, one major difference from a bacteria to our cell is that we have sterol in our cytoplasmic reticulum or cell membrane, whereas bacteria do not contain sterol. Peptidoglycan, as I have said, peptidoglycan in the cell wall is very much important for bacteria, whereas we do not have that. And cellular respiration, in case of bacteria, it occurs via cell membrane, and most of us already know that in case of ours, our cellular respiration or metabolism basically occurs in mitochondria, whereas as they do not have a mitochondria, the cellular respiration occurs in the cytoplasmic membrane. Sorry. And reproduction, we human and animals usually reproduce sexually, and some of other eukaryotes such as fungi or other plants, some of the plants asexually reproduce, but not all, but some of them. And in case of prokaryotes, it's basically asexual or binary fission that we have already mentioned. So moving on to the next portion. So we have already said that the bacteria is prokaryote. Bacteria are independent. So bacteria, we should all be like bacteria because they're free living and independent. They do not depend on anyone else. So, but there are exceptions to that criteria, and that is chlamydia and rickettsia. These are obligate intracellular, and they do need a host to live, so they can be called obligate intracellular parasites. So all the bacteria are free living. They can live on its own without a host. They can survive on crucial environment without any host and without any water. How they do it? They do it by turning into a spore. The sporulation will be discussed in the later part of the class in details. But for now, you just remember that even in extreme heat, even if it's in a desert, even if it's in an icy cold island, they can survive if they turn into a spore. But the exception is chlamydia and rickettsia cannot survive without a host. So what are the different bacterial shapes? So to understand the anatomy or morphology, for example, if you want to understand someone or identify someone, okay, so you have to define the shape of that person, even if we are not trying to be very shallow, but we do notice the shapes of other people, right? So it's not very different when it comes to bacteria. So bacteria can be oval or spherical in shape. It usually cocci or cocci, whatever you want to call it. In some parts they call it cocci, and some parts it's cocci. Either way, it sounds wrong, but yeah. So it can be cocci, like spherical or oval shaped. Then it can be bacilli, or rod shaped. Sorry. Then it can be spirochetes or flexible spiral form. In case of Vibrio cholera, which causes diarrhea especially in the Southeast Asian region, in this South Indian in this India, Bangladesh, Pakistan region, cholera is very much prevalent here. So Vibrio or comma-shaped, spirilla is rigid spiral form, and actinomyces have branching filamentous bacteria. So you can see here, these are the coccus. These are oval shaped. Example, staphylococcus or streptococcus. These are usually oval shaped. Then we have rods or bacilli. What are some examples of rods or bacilli? That is Bacillus anthracis or Clostridium, and these are spirochetes. For example, Treponema pallidum. So as we have already mentioned, Vibrio or is comma-shaped, spirilla, and there is actinomyces, branching or filamentous pattern. So what are the arrangement of bacteria? So for example, some bacteria are found in pairs, some are found in fours like tetrads, or in eight Sarcina. And what does it depend upon? It depends upon the binary fission and tendency of the daughter cells to remain attached to each other. Like you see some of the children, they're so attached to their mom, they cannot just be separated and they just travel in groups. It's sort of like that. So if they're in pair, these are diplococci. Let me just show you the picture. It's easier that way. So these two these two cocci form a diplococci, and the example of it is Neisseria meningitis. These are usually found in diplococci. Then there are tetrads. You can find them in a chain. Then it's a streptococci. You can find them in a cluster. Example, staphylococci. And in groups of 8 is Sarcina. So the anatomy of the bacterial cell. So we are starting from the outside. So some of the bacteria have capsule, but capsule is not necessary for the survival of the bacteria itself. So the outermost layer is the capsule, but it is non-essential, and a lot of bacteria don't have capsule. A lot of bacteria grow up without a capsule. They have a difficult life, but they can survive on their own. So the bacteria that have a capsule is basically they have an extra jacket to protect themselves, and they need the extra jacket because they're weak on them on their self. So they need the capsule for extra help basically. And then we have cell wall. This is from this is from outside to inside this way. So it's basically upwards. So the capsule is the outermost layer, which is non-essential. Then we have cell wall, which is the middle layer, which is very much essential, and the cell membrane, which is very, very much essential, because the cell membrane is where the cellular respiration or the metabolism happens in case of bacteria. So here's a picture. You can see this is the capsule, this is the cell wall, this is the cell membrane, this is the nucleoid right here, but we're not discussing about that. We're discussing about these 3 layers in the following slides. So the essential components. A lot of people will tell you to memorize this, but if you actually understand what everything is and understand the functions of each of these components, you do not need to memorize what are the essentials and what are the non-essential components. Okay. So for example, just let me give you a hint. The cell wall is basically, it's like cell wall is the most unique and distinct quality of a bacteria. So like the peptidoglycan layer in the cell wall of a bacteria is what makes a bacteria, you can say it from the outside, apart from the nucleoid or everything. So the cell wall is very much important. So now comes the cytoplasmic membrane or cell membrane. The cell membrane is responsible for cellular respiration or metabolism, and no one can survive without cellular respiration. So cytoplasmic membrane is important. Ribosome is where all the protein is synthesis. You need that. Nucleoid, you can't survive without a nucleus or DNA. Mesosome is where the tools for cellular respiration is stored, and it is basically an extension of the cytoplasmic membrane. So this is also important. And periplasmic space, where they keep all the goods. That means the bacteria keeps all of its weapon, like protease or hydroxylase or all the good enzymes in this place. So this is very much important for the survival of the bacteria. We'll also elaborate on the functions a little bit more when we go further. And the non-essential components that are not important for survival. They, for example, capsule, the fimbriae or pilus, flagellum, endospore, the granules, and the plasmid. So the cytoplasm is not very different from our cytoplasm, so I'm not going to go into much details. They do not have endoplasmic reticulum or mitochondria, as we have said before in case of bacteria. So just let's move on to the nucleoid. So the nucleoid, in case of bacteria, these are the area of the cytoplasm in which the DNA is located. So the genome it is single circular double-stranded DNA. It is haploid that we have already mentioned before, and it divides by binary fission. And the bacterial nuclei do not have any nuclear membrane, no nucleolus, no histone, no mitotic spindle, which makes it a primitive nucleus that I have already mentioned, and no introns in bacterial DNA. So now let's move on to what is plasmid. So plasmid are basically an extra chromosomal DNA. It's like a spare, you don't really need it, but it's like a spare that you have that you do not want to lose. So all the DNA in case of bacteria are circular and double-stranded. So in case of plasmid, it is also not different. It is circular and double-stranded. They can be present in multiple. They can replicate independently. It doesn't depend on the nucleoid. This is a non-essential component, as we have mentioned before, and it can be transmissible. Let me show you the picture of the plasmid. So you can see in this picture, you can clearly see that the plasmid, this is the plasmid, and it is being transferred from one cell to another, one bacterial cell to another. So you can see there that it can transmit this DNA from one cell to another. This is the plasmid that is lying outside the nucleoid. It is independent. It can replicate independently, and it can be transmitted from one cell to another. So you can see that these can be transmissible, but some of the plasmid is non-transmissible as well. So what is the importance? The major thing that you need to remember, it is that it codes for pathogenesis and antibiotic resistant factors. So what happens that when one bacteria becomes, for example, this bacteria has become they just do not respond to antibiotic anymore. They are like, I outgrew you, you cannot beat me down. So, and they're like, they're also spreading this awareness amongst other bacteria that they have, like, you know what, we have the secret weapon to destroy, to not get affected by antibiotics, and this is the secret copy I'm sending you. So they have coded the antibiotic resistant factor here, and they're sending this secret weapon to another bacteria. So that's how antibiotic resistant factors develop. So through plasmid, so that's why it is very important to know what a plasmid is, though it is a non-essential component, because it can spread the antibiotic resistant factors from one bacterial cell to another. And it is very much used in genetic engineering or biotechnology. So if there is any student who are biotech students, I hope they already know what they're up for. So, okay, before I go into this, I can understand that a lot of things that I'm saying, for this to understand, you need to have a prior knowledge about biology to a certain extent. So I hope the students that are watching this are either have some sort of biology degree, or have taken up biology in their school. So in case of transposon or jumping genes, I just want you to show the picture. The name says it all. So the genes are able to jump from one position to another. So you can see this in this picture, that this is a plasmid, this is a nucleoid, and this is a part of a DNA. This is a DNA that is a part of the plasmid. This is a transposon, and this is a part of the DNA that is part of the nucleoid. This is also a transposon. So in the later pictures, you can see that basically the DNA segment on the transposon from the plasmid has jumped from the plasmid to the nucleoid, and the DNA segment from the nucleoid, the transposon from the nucleoid has jumped from the nucleoid, and then it has shifted its position from one part of the nucleoid to another. So it is jumping. It's constantly jumping. This part has gotten dislodged, has been reattached, and you can see the genes can jump from the plasmid to the nucleoid, or different parts of the nucleoid, or can jump from nucleoid to plasmid again also. So they are basically very good in jumping, people. So what happens is, so what we need to know about this, that they have the ability to move from one position to another. They can jump from one position to another. They can jump from chromosome to plasmid or the reverse. We have seen that. Kudos to them for jumping. So and they can code for what they have. Why is this important? Because they have the code for drug resistance enzymes or toxins or a variety of metabolic enzymes. So basically, they also are like secret weapons that can be transferred from one position to another, like plasmid. They also send the secret weapons for drug resistant enzyme, toxins, and metabolic enzymes. So bad news for us. But one thing that is different from plasmid that they cannot replicate independently, whereas plasmid can. This part has the ability to for undergo mutations in the genes, which actually plasmid can also do. But the only difference from the independent plasmid is that it cannot replicate independently. That's just about it. So moving on. Let us move on. Sorry. So ribosome is not very much different from our cells. Basically, they're also made up of protein. They contain rRNA. The ribosome of ours of the bacteria are 70s, and they are inhibited by certain bacteria. So now let's learn about the granules. So why do we need to know about the granules? Granules are basically the storage for nutrients. They're like a cold store for bacteria. They just store up foods in their fridge. The granules are like that. So some of the granules are polybeta hydroxybutyric acid, glycogen, sulfur, and polyphosphate. But why do we need to know about the granules? Basically, when we are trying to identify bacteria in our labs, and we stain how do we do that? After creating a slide, that we stain them, right? So these granules have the ability to take up the color of that stain. For example, in case of, which is a very common example, is Corynebacterium diphtheria, that it has metachromatic or volutin granules. And when we stain it with either methylene blue or Albert stain, it shows in the slide. For example, you can see in this picture, because of the metachromatic granule, this is Albert stain and this is methylene blue stain. In case of Albert stain, this is the metachromatic granule and it has turned completely dark purple, and this is very much so. If something does that, isn't it very easy to recognize? So thank God for granules. And in case of methylene blue stain, we can see that the granules have taken up a light purple or bluish color in case of methylene blue stain. So this is the importance of granules. So now let's move on to a very important structure of a bacteria, that is the cell membrane. So the cell membrane, structurally speaking, unlike cell wall, which animals do not have, but plants do. But in case of cell membrane, it is actually very similar to any other or normal cell membrane. It is the usual fluid mosaic pattern that we know of, phospholipid bilayer with embedded proteins, a hydrophilic and hydrophobic portion, and proteins embedded within them. But the only thing that we need to remember is that it does not have a sterol. But there's an exception, and that is mycoplasma. Okay, so one thing you need to know about bacteria. Okay, so like in our family, there's this one unruly child who does not just go with the entire family. Okay, that's like a stain, or that's very disobedient, doesn't follow any of the rules. So mycoplasma is basically that son or that daughter of the family, or just that child of the family that doesn't listen to any of the rules that bacteria has laid down upon. So whenever you hear an exception, like bacteria have this and this, but every time the but is mycoplasma. So the exception for every bacterial rules, whenever you hear, it is going to be 80% of the time it's going to be mycoplasma. The rest 20% of the time it's other bacteria that we will discuss in our course. But if you were in an examination hall and you have no idea, if you see an option and the option is mycoplasma, if you do not know the correct answer, go for mycoplasma. Okay, so here is a picture of 1 unit of cell membrane. So the function of cell membrane, we have already said this, but I cannot stress this enough. You gotta remember electron transport and oxidative phosphorylation, or cellular respiration, or metabolism. All of it is done by cell membrane. there can be like thousands of functions, but if you do not remember this, you're doomed, man. So you need to remember this: electron transport and oxidative phosphorylation is the number one function of the cell membrane. Then the rest of them is also important, like secretion of hydrolytic enzymes and proteins, selective permeability, like they are very selective about who they let in and who they let out. So selective permeability and transport of solutes, biosynthesis of carrier lipids on which the subunits of the cell are assembled, and they also bear receptors. So then mesosome. Let's move. We have discussed the cell membrane portion. Now discuss the mesosome. So the mesosomes are basically an invagination of the cell membrane. You can see there is this portion right here is the mesosome, and it is after the invagination, it is continuous with the rest of the cell membrane. And here, this is the principal site for the respiratory enzymes. So, and also you can see that if we pull this invagination towards this side, basically the cell will be divided into two, right? So these also act as an anchor to bind and pull apart the daughter chromosomes during cell division. So you can use this as an anchor and divide the cell. So it's also used as an anchor, you can see here. And also it is analogous to mitochondria in eukaryotes. Why it is analogous to mitochondria? Because you can see that it is basically a specialized form of the cell membrane. Basically, it's an invagination, okay, of the cell membrane itself. So that's why, though the work of the mitochondria is done by cell membrane, but the analogous, if you ask for a form of mitochondria in the bacteria, the answer is going to be the mesosome. Though the function is basically of cell membrane, because analogous, think about analogy. So analogous, like cell membrane is also present in case of in eukaryotes, right? But mitochondria is not present in prokaryotes. So what is the analogous? The mesosome. So this here represents it. So now let's move on to the cell wall. Cell wall is the outermost portion of the bacteria. And as I said, whenever you hear something exceptional, here comes the mycoplasma, the disobedient child. So here's the mycoplasma. So they do not have a cell wall, except for they do not have a cell wall, but all the bacteria do have the cell wall. It's about 10 to 25 nanometer thick. It contains peptidoglycan, which is very much important because like plants also have cell wall, right? But peptidoglycan is very individualistic to bacteria. And they help everything that is a covering. For example, capsule, cell membranal cell wall, they all are going to contribute to the shape and the rigidity, right? So just keep that in mind. And peptidoglycan layer is responsible, and in case of bacteria, this peptidoglycan layer is just so important that it is the main reason for the rigidity of the cell wall. Cell wall cannot be seen in light microscopy or with stain. With simple stains, we can see that with electron microscopes, and the chemical composition is different from bacterial species. So that's how we are going to learn about the bacterial classification in our next class. The classifications are done mostly on basis of this composition of the peptidoglycan layer. So, but we'll just give you some of the gist of that already. Like for example, the cells, the bacteria are mostly divided into gram positive and gram negative, depending on gram stain. Okay, so if we stain a bacteria on a slide with gram stain, so either we are going to find it as a gram positive or a gram negative, right? So this is like one of the different how we differentiate bacteria. This is how, and I'm going to show you the picture in the next slide. So why do we need to know about the cell wall or the peptidoglycan? Because the cell wall is one of the places where the antibiotics attack. For example, the first antibiotic that we know of, penicillin, is basically a cell wall synthesis inhibitor, or cephalosporin. Same, they attack the cell wall of the bacteria. So now let's look at some pictures. You can see that this is the peptidoglycan layer. This is a gram-positive cell wall. This is a gram-negative cell wall. And the peptidoglycan layer is huge in case of gram-positive cell, whereas in case of gram-negative cell wall, the peptidoglycan layer is very much small, and it is basically wedged between these two layers. You can see there is an outer membrane. This is the inner membrane. The inner membrane is basically the cell membrane or the cytoplasmic membrane, and this is the outer membrane. So let me just show you some of the structures that will really help you to understand the later contents with much ease. For example, in case of gram-positive, we can see that the peptidoglycan layer is very thick. There's no outer membrane. Unlike gram-negative, we have teichoic acid and lipoteichoic acid. You can see this is the teichoic acid, this is the lipoteichoic acid. And what is the difference between these two? Teichoic acid is either embedded within the peptidoglycan layer, or it is embedded or it communicates from the outside to the peptidoglycan layer. But the lipoteichoic acid, basically you can see that it is in the peptidoglycan layer, and it is also within the lipid membrane or the cell membrane. So when it crosses the lipid membrane, it is lipoteichoic acid. So this is the gram-positive cell wall, which is pretty straightforward. You can see in case of gram-negative cell wall, you can see there is an outer membrane, then there is a space. This is a periplasmic space, and this is the inner cell membrane, and wedged between these two membrane is peptidoglycan. Now let's learn about the outer membrane. You can see this portion right here. This is lipopolysaccharide. There are different parts of this lipopolysaccharide, and lipopolysaccharide is very important to know because they act as endotoxin, and endotoxin is really bad for us. It causes a lot of problem like septic shock. One of the reasons for septic shock is this portion, this lipopolysaccharide, which is also known as endotoxin. Then we can see these protein channels, which are known as porine. We have this lipoprotein, which basically anchors this peptidoglycan layer with the outer membrane. We have this periplasmic space. In periplasmic space, we have already told you in the beginning. You might not remember. That is the periplasmic space. Basically, have all the secret weapons. This is like a secret weapon storage area. So they have all the good enzymes here, like hydroxylase or peptidase, everything that they need, that they use to destroy their enemies. For example, the macrophage or neutrophils, or the WBCs that we have, or the antibiotics that we use. And this is the cell membrane, which we already have discussed. So what do we need to know about the peptidoglycan layer? Because it's so unique to the bacteria, we need to understand thoroughly what the peptidoglycan layer looks like, right? So first, just let me tell you the difference between the gram-positive and gram-negative staining a little bit. So you can see the major difference between gram-positive and gram-negative was that the peptidoglycan layer, the picture we saw earlier, the peptidoglycan layer thickness is just, there's like a massive difference between the two layers, right? Thickness of the two layers. So that's where the staining basically differentiates. So in case of gram-positive, when we fix it with the slide, then we use crystal violet. You can see it has taken up the color. We treated with the iodine treatment. We can see the purplish change. But when we decolorize it with a little bit of acid, we can see the decolorization. In case of gram-negative, it easily decolorizes, but no decolorization in case of gram-positive. Because of the peptidoglycan layer being so thick, they retain a lot of the stain, a lot of the color, so they do not easily decolorize. Then we use a counter stain with safranin. Because they have not let go of the previous stain, they hold on to the previous stain. But in case of gram-negative, as they have let go of the previous stain, they do take up the newly introduced stain safranin, and they take up this reddish or pinkish color. So that's how we can identify so that's how the gram-negative and gram-positive we can differentiate in a slide. So the chemical composition of the peptidoglycan. So it is within the name itself. You can see the peptide and the glycan portion. So peptide is basically protein, glycan is basically carbohydrate. So it contains so the three parts are: a glycan part is the carbohydrate backbone, the peptide part is the rest of the two, a tetrapeptide side chain, and a peptide cross bridge. Okay, for the next portion, it is going to be a lot of information thrown at you. So, if you need to pause the video and just clear out your head for a while, do that, because it's very important. Especially, it's very important to understand this, because it's going to, because if you want to understand that how the penicillin works on cell wall, you need to understand the composition of the cell wall, and that is basically the peptidoglycan layer. So here we go. Okay, so we learned that there are basically three components. Okay, so the first component was the carbohydrate backbone. So you see this bamboo-like structure, these are basically the carbohydrate backbone. So there are two types of carbohydrate here. You can see there's the light and the dark brown. The light brown is the N-acetyl glucosamine, NAG. The dark brown part is the N-acetyl muramic acid, NAM. These are the reducing substance, or the carbohydrates basically. Now the protein portions. The protein portions were the tetrapeptide side chains and the peptide cross bridges. So you see these are the tetrapeptide side chains, and the proteins that are connecting these two side chains are these cross bridges right here. Okay. So we have learned the names of the carbohydrate. We have not learned the names of the peptides. Okay, so we'll learn the names of the peptide right here. So the peptides are L-alanine, D-glycine, L-glutamate, L-lysine, and then D-alanine, AGLA. Okay, so this is something you have to memorize. I do not have, I cannot say any other way of memorizing, describing it or explaining it so that you'll remember, but you just have to memorize it. This is in case of gram positive. In case of gram negative, this L-lysine portion is actually mesoaminopimelic acid in case of instead of this lysine, but the rest of it is basically the same. And you can see this peptide cross bridges right here, or peptide cross link. This is connecting from this fourth position to the third position. So the D-alanine of one tetrapeptide side chain is connected to L-lysine of another tetrapeptide side chain. So and in case of gram-negative bacteria, this portion, the L-lysine, is basically mesoaminopimelic acid. So in case of gram-negative bacteria, it's going to be D-alanine to mesoaminopimelic acid. Okay. So this is another picture. You can see that, okay, in this picture, one important thing that is shown is that this is this is NAM, this is NAG, you can see, and there's this, in this portion you can see the connection between them, and the connection is called beta 1,4. And this connection is important to know because the lysosome that are secreted from our cells to destroy the bacteria basically works on this, and they break the backbone. When they break the backbone, they break the cell wall. And do you remember if the cell wall is an essential or a non-essential component? Yes, the cell wall is an essential component of the bacteria. So if the cell wall is destroyed, the whole thing falls apart. So we can destroy the bacteria. So this part is basically the part where our lysozyme enzyme works here. What secretes the lysozyme? In most cases, it's the macrophage or the neutrophil. So we have already told you about the tetrapeptide. The name says it all. Tetra means 4. What are the 4 proteins that are alanine, AGLA, as we have said, alanine, glutamine, L-lysine, and D-alanine. In case of gram-negative, this L-lysine portion is mesodiaminopimelic acid. And peptide cross bridges, they connect the fourth to the third, so L-lysine to D-alanine. In case of gram-negative bacteria, D-aminopimelic acid. The enzyme here is transpeptidase. And if this cross-linkage is disrupted, the cell wall becomes defective and cannot protect the cell against osmotic shock, and therefore the bacteria dies. So what is the function? As we have already said here, that when the cross-linkage is disrupted, cannot protect the cell against osmotic shock. So the cell wall protects against osmotic shock. So if there is like more water within the bacteria, or there's less water outside of the bacteria, you can see that they they stop that osmosis from happening, because we know that basically the cell membrane is a semi-permeable membrane, right? But the cell wall inhibits that transfusion, so they act against the osmotic shock. They also maintain the shape of the bacteria. They are a major antigenic determinant, and they're major antigenic determinant because also because in case of gram-negative bacteria, they have lipopolysaccharide. We'll discuss it later. They also provide attachment for the site of a bacteriophage and platform for surface appendages. So the rest of the anatomy of the bacteria, we'll discuss it in the later part. Stay tuned for the rest of it.